Hello there, and welcome. Perhaps today carried more weight than usual, or maybe you simply need a moment to let your thoughts settle like silt drifting down through still water. Either way, you've found the right place. Let your shoulders drop, your breathing slow to something soft and steady, as though you're floating gently just beneath the surface of a calm sea. Tonight we're drifting back through time itself, into the ancient world of crustaceans, creatures whose ancestors crawled through primordial waters long before the first fish learned to swim. These armored wanderers have witnessed the rise and fall of entire worlds, surviving catastrophes that erased most other life, carrying their stories forward in shells and segments that have barely changed across hundreds of millions of years. Some of them are older than mountains, older than forests, older than almost anything alive today. But before we jump in, if you enjoy learning about our oceans, you're welcome to let us know where you're tuning in from and what time it is for you. It's always fascinating to see how far our stories reach, and if these quiet ocean journeys help you unwind, you're always welcome to subscribe so you can drift off with us whenever you need. Now let your body grow a little heavier as we begin. In the beginning there were no crustaceans. There were no shells clicking against rocks in tidal pools, no antenna waving through currents, no segmented legs stepping carefully across the ocean floor. The seas of the early Earth were different from anything we would recognize today, warmer, stranger, filled with microscopic life that had only just begun experimenting with complexity. But somewhere in those ancient waters, more than 500 million years ago, something extraordinary was taking shape. The story begins in a time called the Cambrian period, an era so distant that when you try to imagine it, even the continents are in the wrong places. The world map would be unrecognizable. And yet in those unfamiliar seas, life was exploding into forms more varied and strange than it had ever been before. Scientists call this the Cambrian explosion, a relatively short span of perhaps 20 or 30 million years during which most of the major groups of animals we know today first appeared in the fossil record. Among them were the earliest crustaceans. These first crustaceans didn't look much like the lobsters or crabs you might see in an aquarium today. They were smaller, simpler, with bodies that were still experimenting with the basic blueprint, but already they possessed the fundamental features that would define their descendants for 500 million years to come. They had segmented bodies, each ring of their form capable of bearing a pair of jointed appendages. They had hard outer shells made of a substance called chitin, providing protection in a world where predators were just beginning to evolve efficient hunting strategies. They had compound eyes, or structures that would soon develop into them, capable of detecting movement and light in the murky water. The fossil record from this time, though fragmentary, offers us glimpses of these ancient pioneers. One of the most important windows into the Cambrian world is a place called the Burgess Shale in British Columbia, Canada. Here, in layers of rock that were once the bottom of a shallow tropical sea, paleontologists have found exquisitely preserved fossils that show not just the hard parts of ancient animals, but sometimes their soft tissues as well. It's a rare gift. Usually, when a crustacean dies, its soft internal organs decay quickly, and even its shell often breaks apart before it can be buried and fossilized. But in places like the Burgess Shale, special conditions allowed for exceptional preservation. Among the Burgess Shale fossils are creatures that show us what early crustacean relatives looked like. There's Canada Spice, a small swimming animal with a head shield, segmented body and numerous legs, clearly recognizable as a crustacean even to a modern eye. There's Odaria, with its distinctive tail fan and large eyes. These animals swam through the Cambrian seas, filtering tiny particles of food from the water, scuttling along the muddy bottom, living lives that we can only reconstruct from their flattened remains. But the Burgess Shale isn't the only window into this ancient world. In southern China, there's another remarkable fossil site called the Chengjiang Formation, slightly older than the Burgess Shale, dating back about 520 million years. Here too, paleontologists have found early crustaceans and their close relatives, preserved in such detail that you can sometimes see their antennae, their feeding appendages, even the delicate bristles that help them sense their environment. These fossils tell us that by the early Cambrian, crustaceans had already developed many of the features that would make them successful for hundreds of millions of years. What's remarkable is how quickly this complexity appeared. Before the Cambrian, the fossil record shows mostly simple, soft-bodied organisms. Then, in what seems like a geological instant, though it took millions of years, there's an explosion of diversity, of experimentation with new body plans and new ways of living. Crustaceans were part of this great flowering of animal life, and from the very beginning they were successful. They spread through the oceans, adapting to different depths, different temperatures, different ways of finding food. One group that emerged early and left an extensive fossil record were the ostracods, tiny crustaceans enclosed in a bivalved shell that looks almost like a seed or a bean. These minute creatures are among the first definitive crustacean fossils we have, their small calcified shells preserved well in sedimentary rocks, and we can trace their lineage back through the Paleozoic era. Some of these ancient ostracods were less than a millimeter long, filtering food from the water or scavenging along the bottom. They may have been small, but they were numerous, and their shells accumulated in such quantities that in some places, layers of limestone are composed almost entirely of ostracod remains. The early diversification of crustaceans wasn't limited to one body plan or one way of life. By the Ordovician period, which followed the Cambrian, we see evidence of many different crustacean groups branching out, trying different strategies. Some developed long bodies optimized for swimming. Others became bottom dwellers with flattened forms and legs adapted for walking across soft sediment. Still others evolved into active predators, with grasping appendages and sharp mandibles for crushing and tearing their prey. In Sweden, there are Upper Cambrian rocks that have yielded a treasure trove of well-preserved crustacean fossils. Most of these are small swimming species, captured in fine-grained sediments that preserved even delicate details of their anatomy. Among these Swedish fossils are early ostracods like Vestrogothia and Hässleindona, along with various larval and juvenile stages of unidentified crustaceans that tell us about how these animals grew and developed. There are also representatives of other crustacean groups, showing that by this time diversification was already well underway. What drove this early success? Part of the answer lies in the crustacean body plan itself. The segmented body, with each segment potentially bearing a pair of appendages, is remarkably versatile. Those appendages can be modified for different functions: walking, swimming, feeding, sensing the environment, holding onto a mate, defending against predators. This modular design allows for endless variation. You can add segments, subtract segments, elongate the body, compress it, specialize certain appendages while leaving others generalized. It's a design that evolution could work with, shaping it in countless directions. The hard exoskeleton was another key innovation, made primarily of chitin, a strong but lightweight polysaccharide. The crustacean shell provided protection against predators and support for muscles, allowing for more powerful movements than soft-bodied animals could achieve. Of course, having a rigid external skeleton came with challenges too. To grow, a crustacean must shed its old shell and form a new, larger one, a process called molting. During molting, the animal is vulnerable, its new shell still soft and uncalcified. But this challenge was outweighed by the benefits, and molting became a fundamental part of crustacean life, something that their ancestors evolved more than 500 million years ago, and their descendants still do today. Another advantage was their sensory sophistication. Many early crustaceans had compound eyes, visual organs made up of numerous tiny lenses, each gathering light from a slightly different angle. This design, which also appears in insects and other arthropods, provides excellent motion detection and in some cases impressive visual acuity. Crustaceans could see their world, track moving prey, avoid predators, find mates. They also had antennae, sensitive to both chemical and mechanical stimuli, allowing them to taste the water, detect vibrations and navigate in darkness or murky conditions. As the Paleozoic era progressed through the Ordovician, Silurian and Devonian periods, crustaceans continued to evolve and diversify. We see the first hints of forms that would eventually give rise to modern groups. The Malacostracans, the class that includes crabs, lobsters, shrimp and many others, began to appear in the fossil record. These were more advanced crustaceans, with a more standardized number of body segments and more specialized appendages. They would eventually become the dominant crustaceans in many marine environments, but their rise to prominence took time, unfolding over tens of millions of years. The early crustacean world was a world of experimentation and innovation. Some lineages flourished briefly and then disappeared. Others found successful niches and persisted, evolving slowly or sometimes barely changing at all. The fossil record, though incomplete, shows us snapshots of this long history. A shell here, a fossilized burrow there, occasionally a beautifully preserved specimen that shows us details we never expected to see. Each fossil is a small window into the past, a piece of evidence in a vast puzzle that we're still trying to understand. By the end of the Paleozoic era, about 250 million years ago, crustaceans had established themselves as one of the most successful groups of animals in the ocean. They had survived multiple mass extinction events, adapted to changing climates and sea levels, and spread to virtually every marine habitat. Their basic body plan, refined in those ancient Cambrian seas, had proven itself resilient and adaptable, and the story was far from over. In the ages to come, crustaceans would continue to evolve, to diversify, to colonize new environments, and to weather new catastrophes. But those foundations laid so long ago in the deep past would remain, a legacy written in chitin and stone. There are certain creatures on Earth that seem to carry the ancient past with them wherever they go, animals so old in design, so unchanged from their distant ancestors, that encountering them feels almost like meeting a time traveler. The horseshoe crab is one of these living echoes of prehistory, a creature that has witnessed the rise and fall of the dinosaurs, the formation and breakup of continents, the slow dance of evolution all around it, and yet it has remained, in its essential form, remarkably the same. Despite their name, horseshoe crabs are not crabs at all. They're not even crustaceans. They belong to a different group of arthropods called chelicerates, which makes them more closely related to spiders, scorpions, and ticks than to the true crabs scuttling along the ocean floor. This is a beautiful reminder that evolution sometimes arrives at similar solutions through different paths, and that names given long ago by people trying to make sense of the natural world don't always reflect true relationships. The horseshoe crab's lineage reaches back at least 450 million years, to the Ordovician period when early jawless fish were just beginning to explore the seas. Fossils from this time show animals that already possessed the distinctive horseshoe-shaped shell, the segmented abdomen, the long spike-like tail. When you see a horseshoe crab today walking along the sandy bottom of a shallow bay or emerging onto a beach to spawn, you're looking at a design that has endured for almost 500 million years. It's a humbling thought. What makes the horseshoe crab's form so timeless? Part of the answer lies in that distinctive shell, properly called a carapace, which covers most of the animal's body in a smooth domed shield. This carapace is divided into three main sections. The front section, the cephalothorax, is the largest, shaped like a horseshoe as the name suggests, and it houses most of the animal's organs, its eyes and its legs. Behind this is the abdomen, a smaller hinged section with movable spines along its edges. And finally there's the telson, the long pointed tail that people often mistake for a dangerous stinger but is actually just a tool for steering and for righting the animal if it gets flipped upside down. The horseshoe crab's eyes are perhaps its most remarkable feature. It doesn't have just 2 eyes but 10, scattered across different parts of its body. The 2 largest are compound eyes, positioned on the top of the cephalothorax, each made up of numerous light-sensing units. These can detect movement, and remarkably can increase their sensitivity by up to 10 times in dim light. There are also a pair of simple eyes near the front of the shell, and several more photoreceptors on the underside and along the tail. Some of these eyes are sensitive to ultraviolet light reflected by the moon, helping the animal time its spawning migrations to coincide with high tides during full and new moons. It's an ancient visual system, different from what we find in most modern animals, yet perfectly suited to the horseshoe crab's needs. Underneath the protective shell, a horseshoe crab has 5 pairs of walking legs, each ending in a small pincer, except for the last pair, which has flattened tips for pushing through sand. These legs serve a dual purpose. They're used for locomotion, but they also help with feeding. The horseshoe crab has no jaws or teeth. Instead, the base of each leg is covered with small spines that it uses to crush and grind food before passing it forward to its mouth, which is located in the center of its legs. It's an unusual arrangement, but it works. Horseshoe crabs feed on worms, mollusks, small crustaceans, and other bottom-dwelling prey, crushing them between their legs as they walk. Behind the legs, on the underside of the abdomen, are the book gills, respiratory organs that look like the pages of a book. These gills extract oxygen from water as it flows over them, allowing the horseshoe crab to breathe. Interestingly, these gills can function in air as well, as long as they stay moist, which is why horseshoe crabs can survive out of water for up to 4 days. This ability serves them well during their spawning journeys onto beaches. The blood of the horseshoe crab is one of its most famous features, at least among scientists and medical professionals. It's blue, colored by a copper-based molecule called hemocyan that carries oxygen through the animal's open circulatory system. But what's even more remarkable is what that blood contains. Horseshoe crab blood has special cells called amoebocytes that are incredibly sensitive to bacterial endotoxins. When these cells encounter endotoxins, they clot instantly, isolating the contamination. This is part of the horseshoe crab's ancient immune system, a defense that has protected these animals for hundreds of millions of years. Humans discovered that this property could be used to test for bacterial contamination in medical products. For decades now, a substance called Limulus amoebocyte lysate, or LAL, extracted from horseshoe crab blood, has been used to test vaccines, injectable drugs, and implantable medical devices for the presence of dangerous bacteria. Every time you receive a vaccination or undergo surgery with implanted devices, there's a good chance that horseshoe crab blood helped ensure those products were safe. It's a strange connection between ancient marine life and modern medicine, a reminder that the natural world still has much to teach us. The life cycle of a horseshoe crab unfolds slowly. When they first hatch, the tiny larvae look like miniature versions of adults, though their tail is much shorter. Because of their resemblance to trilobites, those famous extinct arthropods, horseshoe crab larvae are sometimes called trilobite larvae. They grow by molting, shedding their shell and forming a new, larger one. In their first year, they molt about 6 times. It takes at least 9 years and many more molts before they reach sexual maturity. Males undergo a final molt that transforms their first pair of claws into a specialized clasping shape used to hold onto females during mating. When the time comes to reproduce, horseshoe crabs emerge from the deeper waters where they spend most of their lives and journey to the shores. They time these migrations to coincide with the highest tides of the year, the spring tides that occur during full and new moons. On these nights, beaches along certain coasts become crowded with horseshoe crabs. Females drag themselves up onto the sand, each with a male clasped to her shell, sometimes with additional males clustered around hoping for a chance to fertilize eggs. The female digs into the wet sand and lays thousands of tiny green eggs. The males release sperm over these eggs, and then the adults return to the sea, leaving the eggs buried in the sand. These eggs are a crucial food source for migrating shorebirds, especially the red knot, a threatened species that times its northward migration to coincide with horseshoe crab spawning. The birds stop at these beaches to feast on the protein-rich eggs, building up the energy reserves they need to continue their journey to Arctic breeding grounds. It's a relationship millions of years in the making, a connection between ancient arthropod and modern bird that speaks to the interconnectedness of life. The horseshoe crab fossil record is extensive, stretching back through the Mesozoic and Paleozoic eras. Fossils from the Jurassic period, about 150 million years ago, show horseshoe crabs that are virtually identical to modern species in shape and size. Even older fossils, from the Carboniferous and Permian periods, show recognizable horseshoe crab features. Of course, there has been some evolution. Different species have come and gone. Some ancient horseshoe crabs were larger, some smaller. Some lived in marine environments, while others, surprisingly, adapted to freshwater, living in rivers and swamps. Fossils of these freshwater species have been found, showing that horseshoe crabs experimented with different habitats over their long history. But the fundamental body plan has proven remarkably stable. A 300-million-year-old horseshoe crab fossil from the Carboniferous period looks like a horseshoe crab. A fossil from a Polish quarry, dating to the late Jurassic, is nearly indistinguishable from modern species. And a living horseshoe crab, crawling across the bottom of Delaware Bay or the South China Sea, carries that same ancient design. Why has this form endured for so long? Scientists believe it's because the horseshoe crab's design is simply very good at what it does. The hard, rounded shell provides excellent protection. The arrangement of legs and feeding apparatus is efficient. The sensory systems work well. The animal's lifestyle, foraging on the bottom in shallow coastal waters, is one that has been consistently available for hundreds of millions of years. There hasn't been strong evolutionary pressure to change dramatically because the horseshoe crab has always been well suited to its environment. It found a successful way of living very early in its history and stuck with it. Of course, living fossil is a slightly misleading term. No species is completely unchanged. Even horseshoe crabs have evolved in subtle ways. There are 4 living species today, 1 in North America and 3 in Asia, and they differ from each other in small details of size, spine arrangement, and habitat preference. Their ancestors were more diverse, with many species that have since gone extinct. The horseshoe crabs we see today are the survivors, the ones whose particular combination of traits allowed them to persist while others faded away. There's also the fact that horseshoe crabs have survived some of the most catastrophic events in Earth's history. They lived through the Permian extinction, the worst mass extinction event ever, which killed about 96% of all marine species. They persisted through the Triassic-Jurassic extinction and the end Cretaceous extinction that wiped out the dinosaurs. Each time, horseshoe crab populations were probably devastated, but enough survived to rebuild, to carry the lineage forward. That kind of resilience, across such vast spans of time, is extraordinary. Today, horseshoe crabs face new challenges: harvesting for biomedical purposes, collection for bait, habitat loss, and climate change all threaten their populations. In some regions, their numbers have declined significantly. Conservation efforts are underway in many places, with protected beaches and regulations on harvesting. Because horseshoe crabs have been around for so long, it's easy to assume they're invulnerable, but they're not. Like all species, they can be lost if we're not careful. But for now, they persist. On certain spring nights, if you visit the right beaches, you can watch horseshoe crabs emerging from the waves, just as their ancestors did hundreds of millions of years ago. You can see their ancient form, unchanged in its essentials, moving with patient determination across the sand. In those moments, the deep past feels very close, as if time itself has folded, bringing an ancient ocean and a modern shore together in the moonlight. If you've ever walked along a rocky shore, or peered over the side of a dock, you've almost certainly seen barnacles, small volcano-shaped shells clustered together, cemented permanently to whatever surface they've chosen. They seem like simple, even boring creatures, encrusted and immobile, barely recognizable as animals at all. But barnacles are among the most fascinating crustaceans, with a life story that defies expectations and a fossil record that stretches back through hundreds of millions of years. For a long time, people didn't realize barnacles were crustaceans. Their shells looked more like those of mollusks, like limpets or mussels, and they lived attached to rocks and wood like oysters. Carl Linnaeus, the great Swedish naturalist who developed our system of classifying living things, classified barnacles as mollusks. Other naturalists agreed. It seemed obvious: hard shell attached to rocks, filter feeding, that's a mollusk. But in 1830, a British zoologist named John Vaughan Thompson made a discovery that changed everything. He observed barnacle larvae, tiny swimming creatures released into the water by adult barnacles, and he watched these larvae grow and metamorphose. What he saw shocked him. The larvae looked nothing like mollusk larvae. Instead, they resembled the larvae of crustaceans like crabs and shrimp. They had the same basic body plan, the same types of appendages. Thompson realized that barnacles, despite their mollusk-like shells, were actually crustaceans that had evolved a strange and unusual lifestyle. This revelation caught the attention of Charles Darwin, who was then developing his theory of evolution by natural selection. Darwin decided to study barnacles in depth, partly out of genuine interest, and partly because his friend Joseph Hooker suggested that thoroughly understanding one group of organisms would strengthen his theoretical work. What Darwin thought would be a short study turned into an 8-year project that produced 4 monographs describing hundreds of barnacle species, both living and fossil. Darwin's barnacle work remains important to this day, a foundational text in barnacle biology. The barnacle life cycle is complex, divided into distinct stages that seem almost designed to surprise anyone learning about them for the first time. It begins with a nauplius larva, a tiny swimming creature with a simple body and a few pairs of appendages. This nauplius doesn't feed. Instead, it lives off stored energy from the egg. It molts several times, growing slightly larger with each molt, until after about a week it transforms into a different kind of larva called a cyprid or cypris. The cyprid stage is where things get truly interesting. The cyprid has a bivalved shell, like a miniature clam, enclosing its body. It has two large antennules, specialized appendages equipped with sensory structures and adhesive glands. The cyprid does not feed. It has perhaps a few days, maybe a week at most, to find a suitable place to settle. If it fails, it will exhaust its energy reserves and die. The pressure is enormous. During this brief time, the cyprid explores surfaces, walking across rocks, shells, wood or metal with its antennules, assessing each potential home. It's searching for specific cues that indicate a good settlement site. These cues can include surface texture, the presence of certain bacteria or biofilms, and most importantly, the presence of other barnacles, especially members of the same species. Barnacles benefit from living near each other because they reproduce by cross-fertilization, and being close to neighbors increases reproductive success. When a cyprid encounters another barnacle, it detects a protein on the older barnacle's shell called the settlement-inducing protein complex, or CPIC. This protein acts as a chemical signal, telling the cyprid that this is a good place to settle. The cyprid also leaves behind its own chemical traces as it walks, creating footprints of temporary adhesive that contain proteins similar to CPIC. These footprints attract other cyprids, creating a positive feedback loop. Surfaces that have been explored by cyprids become more attractive to other cyprids, promoting gregarious settlement even in the absence of adults. Once a cyprid finds a suitable spot, it commits. Using glands in its antennules, it secretes a powerful adhesive cement, one of the strongest natural glues known. This cement bonds the cyprid permanently to the surface, head first. There's no going back. The animal is now fixed in place for the rest of its life. Then comes metamorphosis, a transformation so dramatic that early naturalists couldn't believe the cyprid and the adult were the same animal. Inside the cyprid's shell, profound changes occur. The thorax, which contains the feeding appendages, rotates 180 degrees. The swimming legs transform into feathery cirrhage, delicate appendages that will be used to filter food from the water. The cyprid's shell is shed, and a new shell begins to form, built of calcified plates arranged in a cone shape. The animal extends upward, building walls around itself, creating the volcano-like structure we recognize as a barnacle. As a juvenile and then an adult, the barnacle spends its life in this self-made fortress, opening and closing a door at the top, extending its cirri through the opening to sweep through the water and capture tiny particles of food. It's a remarkable existence, utterly different from the mobile, free-swimming life of its larval stages. The barnacle has traded movement for security, anchoring itself to one place and letting the ocean bring food to it. The fossil record of barnacles extends back at least 325 million years, to the mid-Carboniferous period. These early barnacles were already recognizable as barnacles, with calcified shells and evidence of the same basic body plan. But paleontologists have found something even more remarkable: fossils that show the cyprid stage preserved in such detail that we can see the antennules, the shell, even some internal structures. In the Wenlock series of rocks from the Silurian period in Herefordshire, England, dating back about 430 million years, paleontologists discovered exceptionally preserved fossils of a barnacle called Rhamphoverita. These fossils show both the free-swimming cyprid larva and a juvenile that had just settled and begun to metamorphose. The cyprid larva is larger than any modern barnacle cyprid, and its antennules end in structures that look almost root-like, different from the attachment structures in modern species. But it's clearly a cyprid, clearly a barnacle larva, proving that this complex life cycle had already evolved by the Silurian. These Silurian fossils are important because they tell us that crown group barnacles, the barnacles that gave rise to all modern species, existed much earlier than scientists previously thought. They show that the barnacle strategy, this unusual combination of mobile larval stage and sessile adult, is an ancient innovation that has proven successful for hundreds of millions of years. Barnacles have diversified into many forms. The most familiar are the acorn barnacles, the cone-shaped ones that encrust rocks and ships. But there are also stalked barnacles, also called goose barnacles, which attach via a flexible stalk or peduncle and suspend their feeding apparatus in the water current. There are barnacles that burrow into shells and coral, excavating chambers for themselves. And there are parasitic barnacles, members of a group called Rhizocephala. which have lost almost all recognizable barnacle features and live as root-like masses inside crabs and other crustaceans, draining nutrients from their hosts and even controlling their behavior to benefit themselves. The evolution of parasitic barnacles is particularly bizarre. The cyprid larva settles on a crab, injects itself into the crab's body through a hollow needle-like structure, and then sheds almost all of its organs, reducing itself to a few undifferentiated cells. These cells grow throughout the crab's body like roots through soil, absorbing nutrients. Eventually, the parasite produces a reproductive sac that emerges from the crab's abdomen and releases larvae. The infected crab is sterilized and often behaves as though it's caring for its own eggs when it's actually caring for the parasite's offspring. It's evolution at its most alien and inventive. The adhesive properties of barnacle cement have fascinated scientists and engineers for years. The cement is secreted as a liquid but quickly hardens underwater, forming a bond strong enough to endure crashing waves, temperature changes, and years of exposure to seawater. It works on all kinds of surfaces: rock, metal, plastic, wood, even the shells of living animals. Understanding how barnacle cement works could lead to new medical adhesives for use inside the human body or underwater construction materials that don't require dry conditions. Barnacles are also economically significant as biofouling organisms. When they attach to ships, they increase drag, reducing fuel efficiency and costing the shipping industry billions of dollars each year. For centuries, people have tried to prevent barnacle attachment with copper sheathing, toxic paints, and other methods. Modern antifouling coatings often work by creating surfaces that are difficult for cyprid larvae to adhere to, or by incorporating compounds that deter settlement. It's an ongoing battle, humans trying to outwit millions of years of barnacle evolution. But perhaps the most beautiful thing about barnacles is simply how improbable they seem. The idea that a swimming crustacean larva would permanently glue its head to a rock and spend the rest of its life kicking food into its mouth with its modified legs feels like something from a fever dream. And yet this strategy has worked for barnacles since at least the Silurian, through ice ages and warm periods, through mass extinctions and the rise and fall of countless other species. The small architects building their tiny fortresses on every shore are proof that evolution finds success in the strangest forms. In the sunlit shallows of tropical reefs among the corals and seagrasses lives a creature that seems designed by a team of engineers with a taste for the spectacular. The mantis shrimp, despite its name, is neither a mantis nor a shrimp, but a member of its own distinct order of crustaceans called Stomatopoda. And it is, without exaggeration, one of the most extraordinary animals on Earth, armed with weapons of devastating power and equipped with eyes more complex than any camera ever made. The mantis shrimp lineage is ancient, branching off from other malacostracans, the group that includes lobsters, crabs, and true shrimp, about 400 million years ago. This was during the Devonian period, often called the Age of Fishes, when early jawed fish were diversifying and coral reefs were beginning to appear. Somewhere in those ancient seas, the ancestors of mantis shrimp developed a body plan that would carry them through eons with remarkable success. What sets mantis shrimp apart? What makes them instantly recognizable even in fossil form are their raptorial appendages.
These are modified limbs,
Typically the second pair of maxillipeds that fold under the animal's body, like the praying mantis arms that give them their common name. But unlike a mantis, which uses its arms to snatch prey, mantis shrimp have evolved these appendages into weapons of astonishing sophistication and power. There are two basic types. Spearers have raptorial appendages lined with sharp spines, used to impale soft-bodied prey, like fish and worms, with lightning-fast strikes. Smashers have club-like appendages, heavily calcified and shaped like hammers, used to batter hard-shelled prey such as crabs, snails, and clams. A smasher's strike is one of the fastest movements in the animal kingdom, accelerating at over 10,000 times the force of gravity, and reaching speeds of around 50 miles per hour in water. The force of impact is so great that it can shatter aquarium glass. The strike creates cavitation bubbles in the water that collapse with a secondary shockwave, strong enough to kill or stun prey, even if the initial blow misses. How did such an extraordinary weapon evolve? The fossil record unfortunately has large gaps, making it difficult to trace the full history of mantis shrimp raptorial appendages. But fossils that do exist provide crucial glimpses. The oldest known mantis shrimp fossils come from the Carboniferous period, over 300 million years ago. These early stomatopods already had raptorial appendages, though they were less specialized than those of modern species. Mesozoic fossils, from the Jurassic and Cretaceous periods, show a range of forms. Some specimens assigned to the genus Sculda, found in Jurassic limestones, preserve remarkable details. Their plepods, the swimming legs under the abdomen, resemble those of modern mantis shrimp. Their antennules and antennae show characteristic stomatopod features, but their raptorial appendages are different. In some Sculda specimens, the first raptorial limb is not significantly larger than the other three pairs. Instead, these appendages decrease in size progressively along the series, more like what we see in some Paleozoic forms. This suggests that the extreme specialization of modern mantis shrimp, with one massively enlarged pair of raptorial appendages, evolved gradually. For a long time, there was a frustrating gap in the fossil record. No mantis shrimp fossils were known from the Permian or Triassic periods, a span of over 100 million years. This gap made it difficult to understand how Paleozoic forms transitioned to the Mesozoic and modern lineages. But recently, paleontologists described fossils from the early Triassic Paris biota of Idaho partially closing this gap. These specimens, named Triassicolda, show a mixture of primitive and advanced features, with a telson shape that resembles Paleozoic mantis shrimp, but a body proportion and spine arrangement more similar to modern forms. It's a transitional picture, exactly what we'd hoped to find. The evolution of smashing versus spearing is another intriguing question. Did mantis shrimp start as spearers and some lineages later evolve smashing capabilities? Or did both strategies evolve multiple times? Molecular studies, analyzing DNA from living species, combined with fossil evidence, suggest that spearing is the ancestral state. The smashing appendage, with its complex structure and specialized strike mechanics, evolved later, probably multiple times in different lineages. This makes sense from a biomechanical perspective. Spearing is relatively straightforward: sharpen the appendage, make it fast, impale prey. Smashing requires more innovation. You need to create a calcified club, develop spring-loading mechanisms to store and release energy, and evolve structures to absorb the massive forces generated by each strike without damaging your own body. Modern mantis shrimp smashers have heel-like structures on their clubs, made of one of the toughest biological materials known. The layers of this material are arranged in a way that prevents cracks from propagating, similar to the design principles used in high-tech ceramics and composite armor. They also have specialized muscles that act like springs, storing energy and releasing it in a burst of acceleration. It's an engineering marvel, refined over millions of years. But perhaps even more remarkable than their weapons are their eyes. Mantis shrimp have compound eyes like other crustaceans, but theirs are divided into three regions: an upper hemisphere, a lower hemisphere, and a midband running horizontally between them. Each eye can move independently, and the tripartite structure allows for binocular vision within a single eye. This means a mantis shrimp can judge distance with one eye alone, something almost no other animal can do. The eyes contain up to 16 types of photoreceptor cells compared to 3 in humans. Some of these receptors are sensitive to ultraviolet light, others detect polarized light waves vibrating in specific planes, which can reveal transparent or camouflaged prey. With this array of receptors, mantis shrimp see a spectrum of colors and patterns invisible to us. For a long time, scientists assumed this meant mantis shrimp had incredibly sophisticated color vision, able to discriminate tiny differences in hue. But recent research suggests their visual system works differently. Instead of processing complex color information in the brain like we do, mantis shrimp appear to use their multiple photoreceptor types as a shortcut, a kind of color barcode system that allows rapid identification of colors without complex neural processing. It's fast, efficient, and perfectly suited to a predator that needs to make split-second decisions. The diversity of mantis shrimp is impressive. There are more than 500 living species, sorted into 7 superfamilies, ranging from tiny species just a few centimeters long to giants exceeding 40 centimeters. They occupy diverse habitats, from shallow reef flats to the deep sea, from sandy burrows to rocky crevices. Some are solitary, aggressive, and fiercely territorial. Others live in monogamous pairs, sharing a burrow for up to 20 years, cooperating to defend their home and raise their young. Mantis shrimp are benthic, spending most of their time on or near the bottom. Many live in burrows which they excavate in soft sediments or find in the cavities of coral rock. The burrow is a retreat, a safe place to hide from larger predators, and a stronghold from which to ambush passing prey. Some species modify their burrows extensively, maintaining them for years, even decorating the entrance with shells and debris. The burrow is also where many species keep their eggs, guarding them until they hatch. Eggs hatch into larvae that drift in the plankton, sometimes for months, before settling to the bottom and metamorphosing into juveniles. During this planktonic stage, the larvae are shaped differently from adults, with enlarged eyes and swimming appendages. They're at the mercy of currents, dispersing across vast distances, which helps explain how mantis shrimp have colonized isolated islands and distant reefs. The evolutionary history of mantis shrimp, pieced together from fossils and molecular data, suggests that the modern lineage called Unipeltata arose around 250 million years ago during the Mesozoic era. This is when the specialized raptorial appendages, both spearing and smashing types, became fully developed. Through the Jurassic and Cretaceous periods, mantis shrimp diversified, experimenting with different body sizes, different hunting strategies, different habitats. They survived the end Cretaceous extinction that killed the dinosaurs, the Paleogene climatic shifts, the ice ages of the Pleistocene. Today's mantis shrimp are the inheritors of this long history, carrying forward adaptations that have proven successful for hundreds of millions of years. They remain important predators in many shallow tropical and subtropical marine ecosystems, playing roles both as hunters and as prey for larger fish, octopuses, and marine birds. In their burrows waiting for prey or patrolling their territory with their extraordinary eyes scanning for movement and color, mantis shrimp are living links to an ancient lineage. They are warriors of the reef, small but formidable, armored in chitin, and armed with weapons that evolution has spent eons perfecting. If you were to catalog all the crustaceans in the ocean, sorting them by diversity, abundance, and sheer ecological importance, you would find that one group stands above the rest, the decapods. These are the crustaceans most people know best: the crabs scuttling sideways across tide pools, the lobsters hiding in rocky crevices, the shrimp darting through seagrass beds. The name decapod means 10-footed, a reference to their 5 pairs of walking legs, though in many species some of these legs are modified for other purposes like feeding or defense. Today there are over 15,000 species of decapods, occupying every conceivable marine habitat and many freshwater and terrestrial ones as well. But this dominance is the result of a long evolutionary journey that began hundreds of millions of years ago. The earliest hints of decapods appear in the Devonian period, about 400 million years ago. Fossils from this time include a creature called Paleopalamin, a shrimp-like animal that already shows the basic decapod body plan, a cephalothorax covered by a protective carapace, an elongated abdomen for swimming, and specialized appendages. Paleopalamin and its relatives were the pioneers, the first to experiment with the decapod design. But decapods remained relatively rare for a long time. Through the Carboniferous and Permian periods, the fossil record shows only scattered occurrences. Other crustacean groups were more common and more diverse. It wasn't until the Mesozoic era, especially the Cretaceous period, that decapods began their great radiation, a burst of diversification that would reshape marine ecosystems. What drove this radiation? Part of the answer lies in the expansion of coral reefs during the Mesozoic. Reefs are complex three-dimensional habitats, full of hiding places, crevices, and surfaces covered with algae and sessile invertebrates. They offer opportunities for many different lifestyles: herbivores grazing on algae, predators hunting small fish and invertebrates, scavengers feeding on detritus, commensals living in association with other species. Decapods, with their versatile body plan and adaptable behavior, were well suited to exploit these opportunities. Fossil sites from the Cretaceous show an explosion of decapod diversity in reef environments. One particularly remarkable site is the Cosco Billow Quarry in northern Spain, which preserves a fossil reef from about 100 million years ago. Paleontologists working here have found 36 species of decapods, including crabs, hermit crabs, and squat lobsters. 8 of these species were previously unknown. This is a record level of diversity for a single Cretaceous site, comparable to the diversity found in modern reef ecosystems. The presence of all the major decapod groups, except true lobsters and shrimp, which may simply not have fossilized well, suggests that by the mid-Cretaceous, reef-associated decapod communities were already complex and well-established. The timing is significant. True crabs, the infraorder Brachyura, which represent one of the pinnacles of crustacean evolution, don't appear in the fossil record until the beginning of the Cretaceous, around 145 million years ago. Once they appeared, they diversified rapidly. An early Cretaceous crab called Telamona carcinus, found in Colombia and dating to about 110 million years ago, is one of the oldest known true higher crabs. Its discovery pushed back the timeline for crab evolution and suggested that crabs originated in tropical regions rather than in higher latitudes as previously thought. Crabs are fascinating because they represent an extreme modification of the basic decapod body plan. In most decapods, the abdomen is large and muscular, used for swimming by rapid tail flips. But in crabs, the abdomen is reduced and folded underneath the body, creating a compact, heavily armored form that's optimized for walking and for defense. This body plan has proven extraordinarily successful. There are nearly 10,000 species of crabs, making them one of the most diverse groups of decapods. The Cretaceous radiation of decapods coincided with the radiation of bony fish, which were becoming more diverse and more efficient predators. Some scientists think that the pressure from fish predation drove decapod diversification, favoring species with better armor, better camouflage, faster movement, or better hiding abilities. The reef environment, with its complexity and variety, allowed many different defensive strategies to coexist. But the Cretaceous was not uniformly favorable for decapods. Near the end of the Jurassic period, there was a major reef collapse, probably due to changing sea levels and climate. When the reefs disappeared, so did many of the animals that depended on them. Decapod diversity plummeted, with over 50% of species going extinct. It was a reminder that even successful groups are vulnerable to environmental change. Yet decapods recovered. As reefs rebuilt in the Cretaceous, decapods diversified again, filling the available niches, evolving new forms. This pattern, collapse and recovery, has repeated many times over the hundreds of millions of years since decapods first appeared. Each mass extinction event winnowed their diversity, but survivors always rebounded, often evolving in new directions, exploring new possibilities. Crayfish, the freshwater cousins of marine lobsters, first appear in the fossil record in the late Permian about 260 million years ago. By the Triassic, they were established in freshwater systems, and by the Jurassic and Cretaceous, they had diversified into many species. Fossil burrows attributed to crayfish have been found in ancient river and lake deposits, showing that these animals have been digging their characteristic tunnels for a very long time. Hermit crabs, which protect their soft abdomens by living in abandoned snail shells, appear in the Jurassic. Squat lobsters, which resemble a cross between a crab and a lobster, also have Jurassic representatives. Slowly, over tens of millions of years, the major decapod groups assembled, each finding its niche, each adapting to different environments and different ways of life. One of the strangest crabs ever discovered is Calichimera perplexa, found in Cretaceous rocks from Colombia and the United States. This crab has been called the platypus of the crab world because it combines features from different life stages in unexpected ways. It has large, paddle-like legs for swimming, more like a larval crab than an adult. It has huge, unprotected eyes, again a larval feature, but it also has adult characteristics. It seems to be a case of neoteny, where some juvenile traits are retained into adulthood. Calichimera was probably an active swimmer and a visual predator, very different from most crabs, which are bottom-dwelling and rely more on chemical cues than vision. The fossil record of decapods has biases. Hard-bodied species with calcified shells are more likely to fossilize than soft-bodied ones. Species living in environments where sediments accumulate, like lagoons and estuaries, are more likely to be preserved than those living on rocky reefs where shells get broken and scattered. This means our picture of ancient decapod diversity is incomplete. We're probably missing many species, especially those that were small, soft-bodied, or lived in environments where fossilization was unlikely. But the fossils we do have tell a compelling story, from humble beginnings as rare shrimp-like creatures in the Devonian. Decapods rose to become dominant members of marine ecosystems. They survived multiple mass extinctions, adapted to changing climates, and colonized habitats from the deep sea to freshwater rivers to dry land. Some decapods, like the coconut crab, live their entire adult lives on land, climbing trees and rarely entering the ocean. Others, like deep-sea lobsters, live in perpetual darkness, navigating by smell and touch in the cold abyssal plains. Today, decapods are among the most economically important crustaceans. Humans harvest millions of tons of shrimp, crabs, lobsters, and crayfish each year for food. They're also ecologically important, serving as predators, scavengers, and prey in food webs. They help recycle nutrients, control populations of smaller invertebrates, and provide food for countless fish, birds, and marine mammals. The great radiation of decapods is ongoing. New species are still being discovered, especially in the deep sea and in tropical regions. Molecular studies are revealing hidden diversity, showing that some species thought to be widespread are actually complexes of closely related but distinct species. The story of decapod evolution is far from finished, and as long as there are reefs and seafloors and river bottoms, decapods will continue to thrive, to adapt, to evolve. Not all crustacean groups that evolved in the ancient past still exist today. The fossil record is full of lineages that flourished for millions of years and then disappeared, leaving behind only their mineralized remains as evidence that they once crawled across seafloors, now vanished into deep time. Among these forgotten groups are some truly remarkable creatures, animals that challenge our understanding of crustacean diversity and remind us that evolution is always experimenting, always exploring new forms. The cyclodons are one such group. These ancient crustaceans first appeared in the Carboniferous period, over 300 million years ago, and persisted until the late Cretaceous, surviving for more than 250 million years. That's an impressive run, longer than the entire age of dinosaurs. And yet today, cyclodons are extinct, and relatively few people have heard of them. Cyclodons looked somewhat like a cross between a beetle and a small crab, with rounded, dome-shaped shells and segmented legs emerging from underneath. The smallest species were just a few millimeters long, their fossils forming what paleontologists describe as looking like tiny bunches of grapes when preserved in clusters. Larger species could reach a few centimeters. For a long time, scientists weren't sure what cyclodons were. They were compared to trilobites, to horseshoe crabs, to true crabs, even to copods. Their classification was uncertain. But in 1997, exceptionally well-preserved cyclodon fossils revealed key details. These fossils showed that cyclodons had two pairs of antennae and front legs adapted for feeding, both characteristic features of crustaceans. This evidence convinced most scientists that cyclodons belong within Crustacea, though their exact placement within the group remains debated. Larger cyclodon species probably filled a crab-like role in their ecosystems, scuttling along the bottom, scavenging or hunting small prey. Smaller species may have been more specialized, perhaps living attached to other animals or hiding in crevices. One of the most intriguing things about cyclodons is how rare it is to find them in groups. Most cyclodon fossils are isolated individuals, suggesting that these animals lived solitary lives, or that their behavior didn't often lead to mass mortality events, where many individuals would be preserved together. But recently, paleontologists in the United States found a remarkable exception, a fossil showing 50 cyclodons gathered together, preserved where they stood over 300 million years ago in what is now Montana. This gathering was buried so quickly, probably by a mudslide during a tropical storm, that the animals had no time to escape. The sudden burial created exceptionally detailed fossils, preserving not just the shells but the positions and orientations of the bodies. By studying these positions, paleontologists concluded that the cyclodons were likely engaged in mass molting. Many crustaceans molt synchronously, gathering together to shed their old shells at the same time. There's safety in numbers. Predators can only eat so many vulnerable, soft-bodied individuals, so if everyone molts at once, more survive. The cyclodon gathering appears to show this ancient defensive behavior, a strategy still used by many modern crustaceans hundreds of millions of years later. Another ancient crustacean group, the filicarids, were among the earliest marine arthropods. Fossils of filicarids appear in the Cambrian period, and they were particularly common in the Burgess Shale and other Cambrian fossil sites. Filicarids had elongated bodies with a bivalved carapace covering part of the body, and they were probably swimmers, using their many legs to propel themselves through the water. One filicarid genus, Canada spis, is among the most common arthropods in the Burgess Shale, suggesting these animals were numerous in Cambrian seas. Phyllocarids persisted for hundreds of millions of years, but their diversity declined over time. By the Mesozoic, they were less common, and eventually they faded from the fossil record. But their importance in the early history of crustaceans is clear. They were part of the initial radiation of crustacean body plans, one of many experimental designs that evolution tried out in those early days. Trace fossils, the preserved evidence of ancient animal behavior rather than the animals themselves, also tell us about early crustaceans. In Texas, in rock layers dating to about 500 million years ago, paleontologists found a trace fossil called Minturichnus, created by a phylocarid crustacean. This trace shows imprints of antennae, walking legs, gill plates, an abdomen, and tail spines, all preserved on the underside of a sandstone slab. The animal that made these traces was about 6 centimeters long and was probably trapped in a tidal pool. This was just before animals first established themselves on land, making it an important snapshot of a transitional time in evolutionary history. The Bredauroids are another enigmatic group from the Cambrian. These small arthropods had bivalved shells and were found in abundance in the Chengjiang Formation of China. For a long time, scientists thought they might be related to ostracods, but more recent studies suggest they might be closer to barnacles. Their exact evolutionary relationships remain unclear, but their presence in Cambrian seas shows that crustacean diversity was already considerable more than 500 million years ago. Some ancient crustacean groups left only fragmentary records. A shell here, a leg there, just enough to know they existed, but not enough to fully understand what they were or how they lived. Others are known from just a few specimens, tantalizing hints of diversity that we'll probably never fully appreciate because the fossil record is simply too incomplete. And then there are groups that we know were diverse and successful, but that left almost no fossils at all. Soft-bodied crustaceans, those without heavily calcified shells, rarely fossilize. If not for exceptional preservation sites like the Burgess Shale, we would have almost no knowledge of Cambrian soft-bodied arthropods. How many other soft-bodied crustacean lineages existed and left no trace? The forgotten lineages of crustaceans remind us that the diversity we see today is just a fraction of the diversity that has existed over the past 500 million years. Most species that have ever lived are extinct. Most lineages have ended. The crustaceans alive today, numerous and diverse as they are the survivors, the descendants of animals that weathered countless environmental changes, extinctions, and competitive pressures. They carry within them the legacy of those forgotten relatives, traces of ancient genes, ancient behaviors, ancient adaptations that once served different purposes in different times. In museum drawers and storage rooms around the world, there are fossils of cyclodens, phyllocarids, bredauroids, and many other extinct crustaceans, waiting for someone to study them, to piece together their stories. Each fossil is a small fragment of a vast, mostly lost history, a reminder that the ocean's past is deeper and stranger than we often imagine. If you could travel back through time and examine crustaceans from different eras, you would see certain features appearing again and again, fundamental aspects of the crustacean body plan that have persisted for hundreds of millions of years. At the same time, you would see endless variation, modifications of those basic features into forms adapted for every imaginable way of life. This combination of conserved structure and innovative variation is what makes crustaceans such a successful and diverse group. The most fundamental feature of a crustacean is its segmented body. Each segment, called a somite, is a modular unit that can bear a pair of appendages. In the ancestral crustacean, all these segments were probably similar, each with a simple pair of legs. But over evolutionary time, different segments became specialized. The front segments fused to form a head, with appendages modified into antennae for sensing and mandibles for feeding. The middle segments became the thorax, with legs for walking or swimming. The rear segments formed the abdomen, which in many crustaceans retained swimming legs called pleopods, or became reduced and tucked under the body, as in crabs. This modular design is incredibly versatile. Want to make a crustacean better at swimming? Enlarge the pleopods, make them more paddle-like. Increase their beat frequency. Want to make it better at walking? Strengthen the thoracic legs, add more muscle. Maybe reduce the abdomen to lower the center of gravity. Want to make it a filter feeder? Modify the legs into feathery cirri that can sweep through water and catch tiny particles. The possibilities are nearly endless. The exoskeleton, the hard outer shell, is another defining crustacean feature, made primarily of chitin reinforced with calcium carbonate in many species. The exoskeleton provides protection and structural support. Muscles attach to the inside of the exoskeleton, allowing for powerful, precise movements. The exoskeleton also prevents water loss, which is crucial for crustaceans that live in intertidal zones or that have colonized land. But the exoskeleton creates a problem. How do you grow when you're encased in a rigid shell? Crustaceans solve this with molting. When it's time to grow, hormonal signals trigger the process. The animal secretes enzymes that partially dissolve the inner layers of its old shell, reclaiming the valuable materials. A new, soft shell forms underneath. Then the old shell splits open, and the crustacean pulls itself out, soft and vulnerable. The new shell expands before it hardens, allowing the animal to grow. Water or air is pumped into the body to increase volume, stretching the new shell to its maximum size before it calcifies. Molting is a dangerous time. Predators can easily catch and eat a soft-shelled crustacean. The animal can't feed, can't defend itself effectively, can barely move. Many crustaceans hide during molting, tucking themselves into burrows or crevices. Some species synchronize their molts, so that most individuals in a population molt at the same time, overwhelming predators with sheer numbers and ensuring that at least some survive. This behavior, seen in the ancient Cyclopean fossils from Montana, has been a crustacean strategy for hundreds of millions of years. The frequency of molting varies. Young crustaceans molt often, sometimes every few weeks, growing rapidly. Adults molt less frequently, sometimes only once a year, or in some species not at all once they reach sexual maturity. Horseshoe crabs, for instance, undergo about 18 molts to reach adulthood and then stop molting entirely. Crustacean appendages show extraordinary diversity in form and function. The baramus appendage, split into two branches, is considered ancestral. One branch is typically a walking or swimming leg, while the other is a gill or sensory structure. But many crustaceans have modified this plan. In decapods, most appendages are ineromous, single-branched, though traces of the ancestral two-branched structure can sometimes be seen during development. Antennae, present in two pairs in most crustaceans, are primarily sensory. They're covered with chemoreceptors that detect dissolved chemicals, allowing the animal to smell and taste the water. Mechanoreceptors detect water currents and vibrations. In some crustaceans, antennae are long and whip-like, constantly probing the environment. In others, they're short and tucked close to the body. Barnacles use modified antennae to attach permanently to surfaces, transforming a sensory organ into an anchor. Mandibles, the jaws, show variation too. In herbivores, they're often flattened and grinding. In predators, they're sharp and cutting. In filter feeders, they may be reduced, since food is collected by other appendages and doesn't need much processing. The shape and strength of mandibles can tell paleontologists a lot about how an extinct crustacean fed. Eyes are another area of innovation. Compound eyes, made of many individual units called ommatidia, are the most common type in crustaceans. Each ommatidium functions as a tiny eye, capturing light from one small part of the visual field. The brain combines the signals from all the ommatidia to form an image. Compound eyes are excellent for detecting motion, but generally don't provide the sharp resolution of vertebrate camera eyes. However, some crustaceans have very sophisticated compound eyes with thousands of ommatidia, providing good detail. A few crustaceans have evolved different eye types. Some deep-sea species have eyes modified to detect the faintest bioluminescent flashes. Others, living in perpetual darkness of caves or the deep ocean, have lost their eyes entirely, relying on other senses to navigate and find food. The carapace, a shield-like extension of the exoskeleton covering part or all of the thorax, is present in many but not all crustaceans. Its evolution and function are still debated. In some groups, like crabs and lobsters, the carapace provides substantial protection and houses the gills. In others, it's reduced or absent. The question of whether the carapace is a primitive feature inherited from early crustaceans or something that evolved independently in different lineages multiple times remains open, though molecular and morphological data are beginning to provide answers. Internal anatomy shows both conservation and variation. Most crustaceans have an open circulatory system, where blood doesn't flow through closed vessels, but instead bathes the organs directly in body cavities. The heart pumps blood through arteries that open into these cavities, and the blood eventually returns to the heart through openings called ostia. This system is less efficient than a closed circulatory system, but is sufficient for crustaceans, many of which have relatively low metabolic rates. Respiration occurs through gills in most aquatic crustaceans. These gills are often associated with the appendages, forming leaf-like or filamentous structures with large surface areas for gas exchange. Water flows over the gills, driven by the beating of specialized appendages. Terrestrial crustaceans like pillbugs have modified gills that function in air, or they've evolved structures analogous to lungs. Nervous systems in crustaceans follow the typical arthropod plan, a dorsal brain connected to a ventral nerve cord that runs the length of the body with ganglia, clusters of nerve cells, in each segment. This ladder-like arrangement is ancient, probably dating back to the earliest arthropods, but there's variation in the size and complexity of the brain, with more behaviorally complex species like mantis shrimp having larger, more intricate brains than simpler filter feeders. The evolution of all these features, the segments, appendages, exoskeletons, eyes, and internal organs, has been shaped by natural selection over hundreds of millions of years. Fossils show us snapshots of this evolution. We can see how carapaces became more extensive in some lineages, how legs became more specialized, how eyes migrated to stalks for better vision. Each change, each innovation represents an evolutionary experiment, tested in the crucible of survival and reproduction. The crustaceans alive today are the descendants of the successful experiments, carrying forward body plans that work, that allow them to find food, avoid predators, reproduce, and pass their genes to the next generation. Fossils are more than just shapes in stone. They're chemical archives, repositories of information encoded in the atoms and molecules that make up their structure. Every ancient crustacean shell carries within it a record of the environment in which the animal lived, the food it ate, the temperature of the water. Paleontologists are learning to read these chemical fingerprints, unlocking secrets that the shape of a fossil alone could never reveal. The process of fossilization begins when an organism dies and is buried in sediment. Over time, minerals from groundwater percolate through the sediment, replacing the original biological materials atom by atom. In some cases, the original structure is preserved in exquisite detail. In others, it's a crude approximation, a mineralized echo of the once living tissue. For crustaceans, the hard exoskeleton is the part most likely to fossilize. Chitin, the organic polymer that forms the base of the shell, can sometimes be preserved, though more often it's replaced by minerals like calcite or silica. Calcium carbonate, which many crustaceans incorporate into their shells for extra strength, is relatively stable and often persists. This calcified shell material is what we find in most crustacean fossils. But the details matter. The ratio of different isotopes of oxygen and carbon in the calcium carbonate can tell us about water temperature and chemistry. Warm water favors the incorporation of certain oxygen isotopes, while cold water favors others. By measuring these ratios, paleontologists can estimate the temperature of ancient seas. They can track climate change over millions of years, see how ice ages cooled the oceans, how greenhouse periods warmed them. Trace elements in the shell provide more clues. Some elements are more abundant in certain environments, and their presence in fossils can indicate whether an animal lived in shallow coastal waters, in the deep sea, in fresh water, or in an estuary. Strontium, barium, magnesium, each tells part of the story. Sometimes, organic molecules are preserved. This is rare, especially in very old fossils, because organic compounds break down over time. But under the right conditions, fragments can survive for millions of years. Proteins, amino acids, even pigment molecules have been detected in fossil shells. These biomolecules can reveal information about the animal's metabolism, its coloration, its evolutionary relationships. One of the challenges in studying ancient crustaceans is that many groups had exoskeletons that were not heavily calcified. Without the reinforcement of calcium carbonate, their shells were more prone to decay and less likely to fossilize. This creates a preservation bias in the fossil record. We have an abundance of ostracod fossils, for example, because their tiny bivalved shells are heavily calcified and remarkably durable. but we have far fewer fossils of some other crustacean groups with more delicate shells. Exceptional preservation sites called Lagerstätten offer a different window. These are places where unusual conditions allowed for the preservation of soft tissues, not just hard parts. The Burgess Shale, the Chenjiang Formation, the Solnhofen Limestone of Germany, the Crato Formation of Brazil. These are just a few of the Lagerstätten that have yielded extraordinary crustacean fossils showing not just shells but muscles, gills, digestive systems, even nervous tissues. In these fossils we can sometimes see things that would normally never preserve: the internal anatomy of a shrimp, the delicate setae on a barnacle's cerri, the gill filaments of an ancient crab. These details transform our understanding. Instead of just inferring how an animal lived based on its shell, we can see the structures it used, the organs that powered its life. Phosphatization, a type of fossilization where calcium phosphate minerals replace tissues, can produce stunning detail. Phosphatized fossils from the Cambrian show embryos of ancient arthropods, larvae at different developmental stages, even cellular structures. These fossils reveal that crustacean developmental patterns, the stages they go through from egg to adult, were already established hundreds of millions of years ago. The chemistry of these fossils can also tell us about diet. By analyzing the isotopes of carbon and nitrogen in fossil tissues, when preserved, paleontologists can determine where an animal sat in the food web. Was it a primary consumer, eating algae and detritus, a predator eating other animals, a scavenger? Each diet leaves a distinct isotopic signature. Metal pore species. Crustaceans, whose shells contain very low levels of trace metals, are typically found in ancient rocks. Well, this makes sense. The early ocean was different from today's ocean, with different chemistry. As life evolved and as geological processes changed the composition of seawater, the availability of different elements changed too. Crustaceans that evolved later, in seas enriched with metals from volcanic activity and erosion, incorporated those metals into their shells. Some fossils preserve evidence of disease or injury: a healed crack in a carapace, abnormal growth patterns, even fossil parasites embedded in the shell. These pathologies tell us that ancient crustaceans, like modern ones, faced threats from predators, from accidents, from infections. They show us resilience, the ability to survive injury and continue living, molting over the damage, sealing wounds with new shell. Coprolites, fossilized feces, occasionally contain recognizable fragments of crustacean shells. These coprolites tell us that ancient predators, whether fish, marine reptiles, or other crustaceans, fed on crustaceans just as modern predators do. Finding a crustacean shell fragment in a fossilized fish turd is a direct, unambiguous record of predator-prey interaction, a snapshot of ancient ecology. Burrows and trackways, trace fossils created by crustacean activity, offer yet another source of information. The shape and size of a burrow can indicate the size of the animal that made it. The branching pattern can suggest whether it was a temporary shelter or a long-term home. The presence of multiple connecting burrows might indicate social behavior. In some cases, the burrow contains the fossilized remains of the burrow maker, giving us both behavior and anatomy together. Reading the chemical and structural fingerprints of ancient crustaceans requires patience, expertise, and sophisticated analytical tools. Mass spectrometers measure isotopic ratios. Electron microscopes reveal microscopic details. CT scanners create three-dimensional reconstructions of fossils still embedded in rock. Each technique adds new information, new insights, new questions. And the work continues. New fossils are discovered every year. New analytical methods are developed. Our picture of ancient crustaceans grows clearer, more detailed, more surprising. Every fossil is a message from the past, written in calcium and carbon, in shape and chemistry, waiting to be read. The ocean was the cradle of crustacean evolution, the place where their ancestors first appeared and where most crustaceans still live today. But over time, some lineages ventured into new frontiers: freshwater rivers and lakes, and eventually dry land. These transitions were not easy. They required profound physiological changes, new ways of breathing, new mechanisms for conserving water, new strategies for reproduction. But the rewards were substantial: access to resources, habitats, and ecological niches that marine crustaceans could never exploit. The horseshoe crab, though not a true crustacean, provides an interesting example. While most ancient horseshoe crabs lived in marine environments, some species transitioned to freshwater. Fossils of these freshwater horseshoe crabs have been found in ancient swamp and river deposits. They show that the transition to freshwater happened multiple times in different lineages, suggesting that it was evolutionarily accessible, though not easy. For true crustaceans, the move to freshwater appears to have begun early. Some Paleozoic crustaceans are found in sediments that formed in freshwater or brackish environments, hinting that the transition may have started hundreds of millions of years ago. But the fossil record for freshwater crustaceans is generally poor. Freshwater environments are less likely to produce fossils than marine ones because they're often associated with erosion rather than deposition. Rivers carry sediment away rather than accumulating it. Lakes can dry up, destroying any fossils that might have formed. Crayfish are among the best documented freshwater crustaceans in the fossil record. They first appear in the Lake Permian, about 260 million years ago, and by the Triassic they were widespread. Fossil crayfish burrows, often called devil's corkscrews because of their distinctive spiraling shape, are found in ancient floodplain deposits. These burrows show that crayfish have been digging for a very long time, creating shelter from drought and predators just as modern crayfish do. Moving to freshwater required adaptations. Freshwater is less salty than body fluids, so water constantly moves into the animal's body by osmosis, diluting its internal chemistry. Marine crustaceans would swell and die in freshwater. To survive, freshwater crustaceans had to evolve mechanisms to actively expel excess water while retaining salts. This is done by the kidneys or kidney-like organs, which filter the blood and produce dilute urine, removing water while reclaiming important ions. Breathing also required changes. Gills work well in water with plenty of dissolved oxygen, but freshwater can have lower oxygen concentrations than seawater, especially in warm, stagnant conditions. Some freshwater crustaceans evolved more efficient gills with larger surface areas or better circulation. Others developed the ability to gulp air from the surface, supplementing gill respiration with air breathing. The transition to land was even more challenging. Air contains plenty of oxygen, but it also dries out gills. Without moisture, the delicate gill filaments collapse and stick together, making them useless. Terrestrial crustaceans had to solve this problem. Some, like beach-dwelling amphipods, never fully left the ocean, living in moist sand or under damp seaweed, running back to the water when threatened. Others, like certain crabs, modified their gill chambers to retain moisture, creating humid pockets where the gills could function even in air. The most successful terrestrial crustaceans are the isopods, the group that includes pillbugs, woodlice, and roly-polies. These small, armored creatures are common in gardens, under logs, in leaf litter. They've evolved structures called pseudotracheae, which are not true lungs but resemble them, tube-like invaginations of the body surface that allow gas exchange with air. They still need moisture and are usually active at night or in damp places, but they're capable of living their entire lives on land. Coconut crabs, the largest terrestrial arthropods, weighing up to several kilograms, have gone even further. They're hermit crabs that have abandoned shells as adults, developing a hardened abdomen instead. They climb trees, crack open coconuts, and only return to the ocean to release their eggs, which develop in the sea before the young migrate back to land. Their gills are modified into branching structures called branchostegal lungs, which extract oxygen from air. To keep these lungs moist, coconut crabs drink water and retain moisture in their gill chambers. Reproduction on land posed challenges too. Crustacean eggs and larvae typically develop in water. Eggs need to be kept moist, and larvae need to swim and feed. Terrestrial crustaceans solved this in different ways. Some, like coconut crabs, returned to the sea for reproduction. Others, like isopods, brood their eggs in a marsupium, a pouch on the underside of the female's body, keeping them moist until they hatch as miniature adults, bypassing the need for a free-swimming larval stage. The fossil record for terrestrial crustaceans is sparse. They're small, often soft-bodied, and live in environments where fossilization is rare. We have few direct fossils of ancient terrestrial isopods or land crabs. Most of what we know about their evolution comes from molecular studies of living species, which allow us to estimate when different lineages diverged and infer when the transition to land might have occurred. But trace fossils help. Burrows in ancient soils, fossilized trackways crossing dune deposits. These indirect evidences show that crustaceans were exploring terrestrial environments millions of years ago. Some of these traces are associated with coastal environments, tidal flats where the boundary between sea and land is blurred, where animals could experiment with life in both worlds. The transitions to freshwater and land were not one-time events but happened repeatedly in different crustacean lineages. Isopods colonized land multiple times. Crabs have multiple independent lineages that adapted to freshwater. This repeated evolution suggests that crustacean body plans are inherently pre-adapted for these transitions, but the necessary modifications are accessible through relatively small evolutionary steps. Today, crustaceans are found in virtually every aquatic habitat, and many terrestrial ones, from mountain streams to desert burrows, from coral reefs to deep-sea vents, from tropical rainforests to Antarctic ice sheets. Crustaceans have proven themselves extraordinarily versatile. The transitions that began hundreds of millions of years ago, when the first crustaceans ventured into fresh water or scuttled onto a primordial beach, opened up new worlds, new possibilities, new chapters in an already ancient story. When you look at a crustacean today, whether it's a lobster on a restaurant plate or a barnacle on a dock piling, you're seeing a design that has been refined over hundreds of millions of years. These animals are not frozen in time. They have evolved and adapted, but they carry within them echoes of their ancient ancestors, features that have proven so successful, so fundamental, that evolution has retained them across vast stretches of time. The segmented body plan is one such echo. From the earliest Cambrian crustaceans to modern lobsters, this modular design has persisted. It's a testament to the versatility of segmentation, the way it allows for specialization without losing basic functionality. A lobster's body is recognizably similar in organization to that of an ancient phylocarid, even though they're separated by over 400 million years of evolution. The compound eye is another ancient feature. Modern crustaceans, from tiny copepods to giant spider crabs, have eyes built on the same basic plan as their Paleozoic ancestors. The details have been refined. The number of ommatidia increased or decreased depending on lifestyle. The spectral sensitivity tuned to different wavelengths, but the fundamental structure remains. It's an eye design that works, that has worked for 500 million years. Molting, too, is an ancient inheritance. The challenges of growing inside a rigid shell were solved early in arthropod evolution, and the molting process used by modern crustaceans is essentially the same as that used by their distant ancestors. The hormonal control, the reabsorption of old shell material, the period of vulnerability, all these are ancient adaptations, preserved because they work. Certain body forms have proven so successful that they've evolved multiple times independently. The crab body plan, with reduced abdomen tucked under a broad, flattened carapace, has appeared repeatedly in different lineages. Hermit crabs evolved a crab-like form independently from true crabs. Porcelain crabs, which are actually more closely related to squat lobsters, also evolved a crab-like appearance. This phenomenon, called carcinization, shows that there's something particularly advantageous about the crab form, some combination of protection, mobility, and efficiency that evolution keeps rediscovering. Living fossils, species that closely resemble their ancient ancestors, are reminders of how conservative evolution can be when a design works well. Horseshoe crabs, as we've discussed, have changed remarkably little over hundreds of millions of years, but they're not the only examples. Some species of fairy shrimp, small freshwater branchiopods, look almost identical to fossils from the Jurassic. Ostracods, those tiny bivalved crustaceans, have forms today that are barely distinguishable from Paleozoic species. This is not to say these animals haven't evolved at all. At the genetic level, there's been constant change. Mutations accumulating, genes duplicating, regulatory pathways shifting. The DNA of a modern horseshoe crab is different from that of its Jurassic ancestor. But the body plan, the morphology, has remained stable because the environment and lifestyle have remained similar. A horseshoe crab living today faces many of the same challenges its ancestors faced: finding food on sandy bottoms, avoiding predators, reproducing on beaches during high tides. The solutions that evolved long ago still work. Other crustaceans show the opposite pattern: rapid morphological evolution. The deep sea, colonized relatively recently in geological time, is full of bizarre crustaceans with strange adaptations. Blind isopods with elongated antennae, spindly-legged pycnogonids, amphipods with bodies modified for life in utter darkness and crushing pressure. These forms are new, evolved in the past few tens of millions of years, showing that crustacean evolution is far from finished. Molecular studies have revealed hidden diversity. Species that were thought to be single, widespread forms are actually complexes of closely related species, each adapted to local conditions. Genetic differences, invisible to the eye, signal separate evolutionary trajectories. This cryptic diversity is especially common in small crustaceans like copepods, where morphology is so constrained by size and lifestyle that different species look nearly identical. Comparisons between fossils and living species reveal patterns. Features that appeared early and persisted are likely to be fundamental, essential to the crustacean way of life. Features that appeared later or that evolved multiple times independently are likely to be responses to specific environmental pressures. By mapping traits onto evolutionary trees, paleontologists and biologists can reconstruct the history of adaptation, seeing when and why particular innovations arose. For example, the evolution of freshwater tolerance can be mapped by looking at which crustacean groups have freshwater species. And when those groups diverged from marine ancestors, scientists can estimate when the transitions occurred. In many cases, freshwater lineages are nested within marine groups, indicating multiple independent colonizations. The same approach works for terrestrial crustaceans, isopods. The most successful land crustaceans belong to a group that includes many marine and freshwater species. By constructing a family tree and looking at where terrestrial species sit on that tree, researchers can infer that the transition to land happened multiple times in different isopod lineages, each time independently solving the challenges of breathing air, conserving water, and reproducing away from the ocean. Modern crustaceans also show us the limits of the body plan. Despite their diversity, there are things crustaceans haven't evolved. True flight, for instance. While insects, which are closely related to crustaceans, conquered the air, no crustacean has evolved wings. Perhaps the constraints of the exoskeleton, the demands of molting, or the aquatic origins of most lineages make flight evolutionarily inaccessible. Or perhaps it's simply that the opportunities for flight haven't been available in the environments where crustaceans live. Similarly, crustaceans haven't evolved the kinds of complex social structures seen in some insects like ants or bees. While some crustaceans are social, living in groups or pairs, maintaining territories, communicating chemically, none have developed the elaborate caste systems and division of labor seen in social insects. Why this is, remains unclear. What modern crustaceans do show us, beyond any doubt, is the power of evolution to shape life over immense spans of time. From simple beginnings in Cambrian seas, crustaceans have diversified into tens of thousands of species, adapted to nearly every aquatic environment and many terrestrial ones, evolved complex behaviors, sophisticated sensory systems, remarkable reproductive strategies, and they've done this while retaining the fundamental features that make a crustacean, the segmented body, the jointed appendages, the chitinous exoskeleton, the compound eyes. These modern echoes of ancient forms are not just curiosities, they're evidence of evolutionary history. Living proof that the past shapes the present, that the designs refined over millions of years continue to serve those who inherit them. And now, as the hours have passed gently by, as you've drifted with me through Cambrian seas and Carboniferous reefs, through the evolution of barnacles and the persistence of horseshoe crabs, perhaps it's time to let the images fade, to let the weight of deep time settle softly around you like a blanket. Crustaceans have been here for so long, longer than mountains, longer than most of the species that share the Earth today. They witnessed the first fish, the first reptiles, the rise of dinosaurs and their fall. They saw continents collide and split apart, oceans open and close. Through all of this, they adapted. They changed when they needed to. They persisted when they could. They survived. Today, they continue that ancient journey. In tide pools and on reef slopes, in rivers and in darkness, crustaceans are living, feeding, molting, reproducing, doing what their ancestors did 500 million years ago. The same basic design, refined and varied but fundamentally unchanged. still works, still allows these animals to thrive. There is something deeply reassuring about this continuity, about the fact that despite everything that has changed on Earth, some things endure. The segmented body, the jointed leg, the compound eye. These features have outlasted empires and ice ages, mass extinctions and tectonic upheavals. They will likely outlast us. In your own life, perhaps there is a lesson here, in the resilience of simple, well-tested designs, in the value of adapting without losing what makes you fundamentally yourself. But perhaps that's too much thinking for now. Let those thoughts dissolve. Let the weight of your body sink deeper into rest. Somewhere tonight, in an ocean you may never see, a larval barnacle is drifting in the plankton, searching for a place to settle. A mantis shrimp is waiting in its burrow, eyes scanning the darkness. A horseshoe crab is walking across the bottom, ancient and patient, following paths its ancestors walked before the first flowers bloomed. The world they inhabit is the same world we inhabit, the same blue sphere spinning in space, the same ocean that has always been here in one form or another, holding these stories, these lineages, these quiet victories of survival. Rest now, let the deep time of crustaceans carry you. Let the knowledge that life is old and strange and resilient wrap around you like warm water. You are part of this same story, made of the same atoms that once formed Cambrian seas, connected by ancestry to everything that has ever lived. The ancient crustaceans swim on through time, and you drift gently with them into the quiet darkness, into sleep. Good night.